Hi, my name is Caitlin, and I'm a graduate student in Dr. Catherine White's lab at the University of Notre Dame. Today, I'd like to discuss a little bit about my recent project that was published in JACS. Intercellular pH, or pHi, is highly regulated in normal cells, and transient increases in pHi have been linked to a variety of pH-dependent cell behaviors, including cell polarization, directed cell migration, and cytoskeleton remodeling. However, the studies linking increased pHi to these pH-dependent cell behaviors were done at the population level using non-specific pHi manipulation methods. The non-specific nature of these tools make it difficult to pinpoint the exact role that increased pHi is having on driving cell behaviors. In order to better understand this relationship, we need new tools to increase pHi that are specific and spatiotemporally controlled. In this work, we present the use of archirhodopsin, or Archi, which is a light-driven outward proton pump, as a tool to spatiotemporally increase pHi in single cells. We paired this tool with the genetically encoded pH biosensor M-cherry-fluorine. The co-expression of Archi and M-cherry-fluorine allows us to manipulate and monitor pHi increases in single cells in real time. We characterized Archi as a tool to increase pHi and showed that Archi could be used to increase pHi within a physiological relevant range, similar to those reported during pH-dependent cell behaviors, and that Archi can be used to repeatedly increase pHi for approximately 45 minutes. Next, we wanted to apply Archi to investigate the role of increased pHi in driving membrane ruffling. For this experiment, we had two hypotheses, both supported by literature. The first is that we would see global membrane ruffling responses as protons diffused from the stimulation region across the cell and pHi increased across the entirety of the cell. The second is that we would see localized membrane ruffling responses as pH-dependent proteins were recruited to the stimulation region and reinforced membrane ruffling events. To our surprise, we found that when we activated Archi and increased pHi, we actually saw localized membrane ruffling responses. This suggests for the first time that increased pHi can drive localized membrane ruffling responses. This project has a wide variety of applications. With this tool, we can begin to specifically investigate the role of increased pHi, both in normal cell behaviors and in the development of disease states such as cancer, where pHi has been shown to be constitutively increased. The development of first-generation tools to increase pHi allows us for the first time to begin to elucidate the molecular mechanisms driving both normal cell behaviors and cancer cell behaviors, as well as determining when and where pHi increases are required during pH-dependent cell behaviors.